Today, we are reporting from the 2020 ESMO Virtual Congress. We're recapping some of the top news that are being presented during the meeting, and in just a bit, we'll be speaking with Dr. Scott Tagawa to see what he thinks about some pivotal genitourinary cancer studies, specifically with bladder cancer and prostate cancer, and also with Dr. Stephen Liu on the biggest ESMO abstracts in lung cancer. Welcome to OncLive News Network. I'm Gina Morrow. In breast cancer, the addition of tamoxifen to abemaciclib resulted in an improvement in overall survival compared with abemaciclib monotherapy in patients with hormone receptor positive, HER2-negative metastatic breast cancer, with benefit observed across all patient subgroups, according to results from the final OS analysis of the next MONARCH trial. In the phase 3 ASCEND trial, treatment with sacituzumab govitecan led to a 59% reduction in the risk of disease progression or death compared with physician's choice of single-agent chemotherapy in patients with previously treated metastatic triple-negative breast cancer. The median progression-free survival by blinded independent central review was 5.6 months in the sacituzumab govitecan arm, compared with 1.7 months with standard treatment. Sacituzumab govitecan also nearly doubled the median overall survival at 12.1 months compared with physician's choice of therapy at 6.7 months, an improvement that was determined to be statistically significant. Also in breast cancer, data from the final overall survival analysis of the SOLAR-1 trial showed that it did not cross the pre-specified O'Brien-Fleming efficacy boundary in postmenopausal patients with PIK3CA mutant, hormone receptor positive, HER2-negative advanced breast cancer. The trial looked at alpelisib and fulvestrant in this patient population, which did prolong the median OS by 7.9 months versus fulvestrant alone, and additionally suggested a survival benefit in various patient subgroups with no new safety signals. In colorectal cancer, frontline pembrolizumab demonstrated clinically meaningful improvements in the health-related quality of life of patients with microsatellite instability high and/or mismatch repair deficient metastatic disease compared with standard of care chemotherapy, as seen in an analysis of the KEYNOTE-177 trial. In bladder cancer, sacituzumab govitecan continued to showcase significant activity with favorable tolerability in heavily pretreated patients with metastatic urothelial carcinoma who progress on both platinum-based chemotherapy and checkpoint inhibition, according to final results from Cohort 1 of the Phase 2 TROPHY-U-01 trial. The overall response rate was 27%, including a complete response rate of 5% and a partial response rate of 22%. In renal cell carcinoma, longer follow-up of the CheckMate 214 study showed the benefits conferred by the frontline combination of nivolumab and ipilimumab for patients with advanced disease remained durable after 4-year follow-up compared with treatment with sunitinib. Investigators also reported that a subgroup of patients with one or more target kidney lesions experienced kidney tumor reduction and other benefits from the combination treatment. In lung cancer, findings from the Phase 3 CROWN trial showed that first-line treatment with the third-generation ALK-TKI lorlatinib significantly improved progression-free survival and also was associated with higher overall and intracranial response rates compared with crizotinib in patients with ALK-positive non-small cell lung cancer. In endometrial cancer, dostarlimab showcased durable anti-tumor activity in patients with advanced or recurrent DNA mismatch repair deficient and proficient disease, with a notable disease control rate and a promising safety profile, according to updated results from the Phase 1 GARNET trial that were presented during the meeting. For more coverage of the 2020 ESMO Virtual Congress, please be sure to visit OncLive.com. We are here with Dr. Scott Tagawa, who is a professor of medicine and urology at Weill Cornell Medicine and an attending physician at New York Presbyterian Weill Cornell Medical Center, to discuss some of the pivotal genitourinary cancer abstracts that are being presented at this year's meeting. Hi, Dr. Tagawa. How are you? It's so nice to see you virtually at the Congress this year.
I'm doing great. Thank you very much for the invitation. I think it's very nice as we get more used to these virtual conferences. And this one's a little bit more interactive, at least through some of the presidential symposiums yesterday.
I've been hearing that a lot, that it is a very interactive meeting this year. So we're definitely refining the virtual conference platforms as time goes on during COVID, I guess, right?
Yeah.
So this year, I'd really like us to focus in on some of the bladder cancer and prostate cancer studies that are being presented. Let's first start with the final results of Cohort 1 of the TrophyU-01 study with sacituzumab govitecan. I know that these are some really interesting data this year that a lot of folks in the community are talking about.
Sure. So sacituzumab govitecan is an antibody-drug conjugate with the target of TROP2. TROP2 happens to be overexpressed in a number of different solid tumors, in particular urothelial carcinomas, the vast majority. So all the studies to date in urothelial have been without target selection. There was a phase 1 trial that included a basket arm of urothelial that looked interesting, about a 30% overall response rate. It was an earlier era, mostly prior to immune checkpoint inhibitors. So the TrophyU-01 study was designed as a dedicated study in patients with metastatic urothelial carcinoma after platinum chemotherapy and immune checkpoint inhibitors, at least Cohort 1, which is being presented. It was designed as a Simon 2 stage. The first stage was presented last year at ESMO with positive results. So we proceeded with stage 2, and these are the primary results of Cohort 1, which was in that refractory group. As it turned out, this was a median of 4th-line therapy for the patients with refractory disease. Majority had visceral disease, although most of that visceral disease was in the lung. And the bottom line is that the results were more or less replicated. So the overall response rate by RECIST was 27%, which is favorable compared to what we expect with a historical control of around 10%. In addition, it was fairly well tolerated. So we know from a number of different studies with hundreds of patients across different diseases that myelosuppression is dose limiting, and that happened as well. Febrile neutropenia was a little bit less in this expanded cohort than it was at the presentation last year. We're still working on the details, but it does look like there were fewer patients with UGT1A1 polymorphisms in this overall group, more of a normal patient population. And then diarrhea also is relatively common, but usually grade 1 or grade 2. The grade 3 rate was low overall. This particular drug did receive fast track designation by the FDA. So this study is potential to lead to accelerated registration.
Thank you so much. Those are some great points about the data. Now, we finally saw the approval of sacituzumab govitecan in breast cancer earlier in 2020. So if we do get approval in urothelial cancer, how do you think that this is going to impact clinical practice and how it can be used when trying to determine sequencing decisions?
So likely it will initially be late line as per the study. So we know that we have platinum-based chemotherapy that remains standard through studies that were also presented at this conference. Now with the introduction of switch maintenance to avelumab or potentially pembrolizumab, so that all that combination or frontline immune checkpoint inhibitors, which has now become a little bit more controversial in my mind, but in any case, frontline is well, that would change this year, but that remains the standard. There's a select group, maybe 1 in 5, that will have erdafitinib as an option. And then EV received accelerated approval at the end of 2019, and that remains with the recent press release of their positive randomized data against quote standard chemotherapy, taxanes, or vinflunine. So I'm interested to see those data, but I would expect that the average patient would receive some combination of those drugs in some sequence as a general rule. One of the nice things about SG, at least in the world of having EV approved, is that it's a different target, a different toxin. So we'd expect there to not be a significant cross resistance, and with very small numbers, it looks to be true. So about 30% response rate of SG following EV, which is more or less the same as it is in those without EV. So very small numbers, but it's nice to see that. And especially nice is that there's generally speaking, not the same toxicity profile. So if someone happened to have neuropathy following platinum and EV, that generally is not an issue with trastuzumab govitecan. So anyway, to answer your question, I expect it to initially be later, but as is very enjoyable and rewarding for me, continuing to do clinical trials with a number of different agents and moving them forward. So for instance, EV is now being studied in a randomized trial in the frontline platinum cisplatin ineligible population, and there's there's additional combinations in the frontline setting with that drug, and even neoadjuvant. So as drugs like immune checkpoint inhibitors move up to the frontline setting, it moves what was previously third line to the second line, and that will kind of continue to happen. The Trophy User 1 study, Cohort 3, is just starting, but that was presented by Johan yesterday in one of the introductory slides, which is similar to EV plus Pembro, except as SG plus Pembro in the frontline cisplatin unfit population. So it'll be interesting to see those data as well.
That's great. There's so much really interesting trials going on, and a lot of very interesting recent data that have been read out recently too, with all these different targeted agents, antibody drug conjugates. There's a lot going on in bladder cancer. Now, you did also bring up avelumab, and I do want us to talk about the analyses that we're seeing from the Javelin Bladder 100 trial, looking at avelumab as frontline maintenance in urothelial cancer. So we saw a mix of data being presented at this year's meeting. Could you highlight on those findings that we saw?
Yeah, I thought that was very interesting, but mostly hypothesis generating. So one thing to remember about that particular trial is that those that had primary refractory disease were not entered into that trial. So perhaps we're not able to look at true resistance markers because they were not entered into that trial, although we have plenty of others in kind of the initial data sets for these drugs, which is platinum refractory, where everyone got immune checkpoint inhibitors. But anyway, that's one comment. Looking at PD-L1 and tumor mutational burden, it looks like, as in most trials, there is some signal that is there, but certainly not everything. So that was nice to see it as kind of confirmatory, both in terms that there looks to be a directional association with those biomarkers, but they don't tell the entire story. So that's also confirmatory that there's always been, there's people that don't respond that should, and vice versa. Well, the response is not the exact answer in this one because it was really overall survival following response or stable disease to chemotherapy. But in any case, the next set of biomarkers, which were different signatures, that I found very interesting, but even more hypothesis generating. But it was very nice for me to see as a non-investigator in that particular trial, for them to collect the samples and do that work, I think is quite important. So I always, like to participate in studies that will embed biomarkers either as a main primary secondary endpoint or exploratory hypothesis generating, and that's how we really learn about patient selection, particularly as we have a growing number of options, and that's becoming really important.
Absolutely. And the other analysis too, looking at how it really benefited across a lot of different patient subgroups that were involved in that trial, really just interesting confirmatory data.
Correct. Yeah, there's, I'd say the average patient that has metastatic urothelial carcinoma receives platinum chemotherapy, the average patient should receive maintenance.
Absolutely. Thank you so much. Now, obviously, there are additional bladder cancer studies that are being presented during this meeting. What other ones would you like to highlight?
Well, those that you partly covered with Shilpa Gupta yesterday, but there were two studies presented at the first presidential symposium yesterday. One of them I am pretty sure that you did cover with Shilpa, a brief on that, the Keynote 361 study. So another study that basically says it's hard to really beat platinum chemotherapy. So the addition of pembrolizumab did not improve over cisplatin or carboplatin plus gemcitabine. The Danube study, I don't think you fully covered. So that was a 3-arm study that was looking to either replace platinum chemotherapy, so a PD-L1 plus a CTLA-4 inhibitor, so durvalumab plus tremelimumab, versus platinum-based chemotherapy, or there was another arm, so each one was separately compared to platinum-based chemotherapy, durvalumab, the PD-L1 inhibitor, in a PD-L1 selected population, went head-to-head against chemotherapy. And the bottom line that neither were overall positive. So that really cements the prior data that we knew from prior presentations as well as press releases, that frontline platinum-based chemotherapy is really hard to beat. That's the way I'd spin it. So that remains the standard. Luckily, we have the results that we just mentioned with the Javelin data and the phase 2 pembrolizumab data that maintenance immune checkpoint inhibitor does improve outcomes. So that should be the standard of platinum-based chemotherapy, followed by, rather than combined with, an immune checkpoint inhibitor. The data set, the data sets in total, with semi-positive data from Atezo, in my mind, really cement platinum chemotherapy. I think we need to see a little bit more information about, for someone that is cisplatin ineligible but carboplatin eligible, and happens to be PD-L1 high. What about carbo gem, for instance, or pembrolizumab or atezolizumab? That's an unanswered question. My view, in a kind of high level initial impression, is that those with disease that is symptomatic and progressive probably should get gem carbo because of for a population, a faster response rate, and then they can have the benefit of maintenance of avelumab. But those with maybe slow growing disease that are PD-L1 high might still be able to get pembro or atezo. That we'll wait to see when hopefully someone's able to do a meta-analysis.
That's great. Thank you so much. Really, really great information here. Now, I do want to move on to prostate cancer as well. There are some great studies here to talk about. Let's first talk about the Epotential 150 trial. That's a phase 3 of episertib plus abiraterone versus placebo plus abiraterone in metastatic castration-resistant prostate cancer. What are your thoughts on these data?
So, a highline is always, anytime that I can have another drug that might benefit some of my patients, that's always great to see. So we don't have approval of this drug in prostate cancer, but this study has the potential to lead to approval. So there was a randomized phase 2 of this drug in combination with abiraterone that happened to be in a docetaxel refractory population, where there was a signal, particularly in the group that had PTEN loss in their tumors. And that led to this randomized phase 3 study in the frontline metastatic CRPC setting of abiraterone prednisone alone, or abiraterone prednisone plus ipatasertib, which is an AKT inhibitor. The study design looked at all comers, as well as prospectively had a subgroup to look at those specifically with PTEN loss. And there was a splitting of the alpha for that. So overall, it looks like there's some benefit for all comers, but really what was statistically significant is an improvement in radiographic progression-free survival in those with PTEN loss in tumors that was designed to be a simple protein immunohistochemistry test, but by next generation sequencing that looked like that was also can be done, although that was more of a secondary exploratory endpoint. So the bottom line is that we have potentially another drug. It is one that scientifically makes sense. It's one that's built upon a randomized phase 2 trial It's another one that is in a biomarker selected subset. So as we continue to move forward in most tumor types, not just within GU, but in most tumor types with precision medicine, this is an exciting new drug. As like we talked about with urothelial, as drugs move forward. So for instance, abiraterone use in the castration-sensitive population, what does that mean? Can we add this drug at progression to castration resistance? Can we extrapolate this to other AR pathway inhibitors? And there's a little bit of data with that. We don't know, but in my mind, it will be very nice to have. Hopefully, we'll have this in the future as a combination strategy. Forgot to mention, there was more toxicity associated with the addition of another drug, which would be expected. Some of this on target, for instance, with hyperglycemia. But in my mind, most of the low grade and the benefit that was seen, in my mind, outweighs the adverse events.
Great. Thank you so much. These are really, really interesting points to bring up. Now, we also saw the approval of a Laparib for patients with mCRPC who have homologous recombination repair alterations in May. Now we're seeing the final overall survival data from the PROfound trial. So if you can shed some light on these findings, and how do you think PARP inhibitors are going to continue to evolve the field of prostate cancer?
So, most of us expected that Laparib was going to get FDA approval, and it did a few months ago. But most of us were curious, was this going to be in BRCA1, 2 ATM, BRCA2 only, or across the board in those selected patients with DNA repair defects? And it turned out it was fairly broad, which in my mind makes it much better for patients to have those options. The patient population, besides being selected for those with DNA repair defects, was post potent AR pathway inhibitors, with or without prior chemotherapy. So that's the patient population that is on label that I think is most appropriate as we study earlier disease states in either selected patients with the drug alone, or the PARP inhibitor alone, or maybe in unselected with different combinations. But in any case, getting to the PROfound data, so we saw the presentation and publication of the RPFS population that was positive in overall and in the different subsets, mostly, in my opinion, driven by BRCA2. But there are other data sets or other subsets that also look like they benefit over dealer's choice AR pathway inhibitors. We also knew there was a press release of overall survival data, and that's what we're seeing at ESMO 2020. So not surprising, since we already knew about that, but the more data that are in the public domain, looking at the different subsets, the better. So this doesn't change things so much for me because we already have approval based on RPFS, but anytime that we have what we consider the more important endpoints, such as overall survival, patient-reported outcomes, if it's done well, is also very important. But it's very nice to see that this led to an overall survival benefit despite crossover in other drugs. So it gives me hope as we move these drugs forward in terms of lines of therapy, that there will also be a benefit. Again, like in the Epotential study, in a selected patient population, which it's not the older days where we have ADT and then we finally had a taxane, and we chose between a taxane or taxane for metastatic CRPC. So biomarkers were a lot less important. Now that we have these different treatment options, these are becoming even more important.
Absolutely. Now, there are obviously so many other prostate cancer studies being presented. We're seeing some early data tomorrow with the AMG 160, the BITE in metastatic castration-resistant disease. What other kind of studies, or if you'd like to shed light on that one as well, would you like to highlight that are being presented at this year's meeting?
So, broadly speaking, as you may know, prostate-specific membrane antigen, or PSMA, is one of my favorite targets. And as I talk to different patients or colleagues, we can use that diagnostically in terms of imaging, and that hopefully is becoming more available in the United States. We can also use it therapeutically, either attaching a drug, with an antibody-drug conjugate or a small molecule with a toxin on it, with radionuclide, and we await results from the VISION trial following positive results of randomized phase 2, but also as an immune target, whether it's a CAR T cell or more of a BiTE. We saw at a couple of conferences results of AMG 212, which was also called different compounds before, which is a little confusing, but a PSMA CD3 bispecific that has a short half-life, so it's continuous infusion. But what we saw is the potential for long-term benefit. And then this year, a couple of BiTEs, either a construct from Harpoon that adds an albumin binding component, so it leads to a longer half-life, so the continuous infusion administration is not necessary, or Amgen's version, AMG 160, which is being presented at ESMO 2020, of essentially changing the size to make it a half-life extended, that can be administered every 2 weeks. So, it's still early data with these drugs. We do know that from blinatumab, the approved version, in hematologic malignancies, that there can be cytokine release syndrome. We do see that with these drugs, although with different ways of administration, titrating up, it looks like that can be improved. And we've learned from our experiences. I talk to my leukemia colleagues and say, hey, I'm going to need your help as we treat these patients. And so as prostate cancer docs learn to handle those, I think it'll become safer. So anyway, as we learn about tolerability, why we really like immunotherapy across the board, we talked about it with urothelial cancer, and now we're getting there in prostate cancer, I hope, is the potential for a durable response. The early data is early data, so we don't know if any of the responses are durable, but we know that they might be, and these drugs are being looked at alone or in combination with other drugs, such as an immune checkpoint inhibitor. So broadly speaking, I think PSMA is becoming a therapeutic target that is really being validated. And at ESMO 2020, we see one of the immune mechanisms of a BiTE, a second or third one, depending on how you look at it. And there's others that are also in development. So I think that the target is a good one. And it looks like we're learning to handle the toxicity profile that will hopefully translate into our ability to administer the drugs and lead to durable response.
There's so much going on. It's so exciting to hear about all of this. Dr. Tagawa, thank you so much for sharing everything today. It was really interesting to get your insight on the bladder cancer and prostate cancer studies, some of the key data being presented at this year's meeting. Thank you so much for taking the time out of your busy schedule, especially during ESMO weekend.
Thank you very much.
We are here with Dr. Stephen Liu, who is an associate professor of medicine and director of thoracic oncology and director of developmental therapeutics at the Georgetown Lombardi Comprehensive Cancer Center, to discuss some of the very exciting lung cancer abstracts that are being presented at the virtual 2020 ESMO Congress. Hi, Dr. Liu. How are you? It's so nice to see you. I wish it was more in person, but it's nice to see you over Zoom.
Yeah, same here. I certainly wish that we were both in Madrid right now.
Yes.
We can make do with where we are.
Yeah, absolutely. So it is really great to be able to sit down with you and talk through some of these really pivotal lung cancer abstracts that we're learning about at this year's meeting. Let's maybe dive into some of the targeted therapies first. The CROWN trial was a very highly anticipated study looking at frontline lorlatinib with crizotinib in ALK-positive NSCLC. So could you perhaps walk us through lorlatinib's timeline and what we saw with this trial?
Yeah, this is an important study. Now, lorlatinib is a highly CNS penetrant kinase inhibitor. It's got activity at ROS1 and at ALK. It was initially approved for use after progression on prior ALK kinase inhibitors. It has demonstrated excellent CNS activity and an ability to overcome a lot of the known ALK solvent front mutations, with responses seen even after multiple kinase inhibitors. The CROWN trial, presented at ESMO by Ben Solomon, was a randomized phase 3 trial for patients with treatment-naive ALK-positive lung cancer. In this study, patients were randomized to receive lorlatinib, 100 milligrams a day, or crizotinib, 250 milligrams twice a day. Now, already, many have noted that our current standard of care is not crizotinib. It is alectinib or brigatinib, which makes interpretation of these data and integration into our own practice a little challenging. I'll remind folks that CROWN was launched in 2017, prior to the approval of alectinib in this setting, certainly prior to the approval of brigatinib. And when you launch a phase 3 trial, your comparator really does have to be the current standard of care. So over these past few years, we knew that lorlatinib would be better than crizotinib. It's really just been a matter of how much better. What we saw at ESMO 20 was that it was much better, with a PFS hazard ratio, a staggering 0.28 by blinded independent review. We compare that for reference to 0.47, 0.49 that we see with the second generation TKIs. The intracranial response rate, sort of one of the big features of lorlatinib, was 82% with a 71% complete response rate. This is important because ALK-positive lung cancer is a significant tropism for the brain, and neuroprotection is very relevant. So the intracranial time to progression was much better with lorlatinib, with a hazard ratio of 0.07, probably the lowest I've ever seen in the trial. You know, we always balance efficacy with toxicity as well. And something to keep in mind is lorlatinib has some unique toxicities, hyperlipidemia, which can be managed with certain statins. But more concerning to us were the potential neurocognitive effects of lorlatinib, which affect memory, clarity, cognition, mood. You know, these are subtle. I've seen these myself. They are concerning, but if we recognize them, they respond well to dose modification. So I certainly don't consider them insurmountable. And the discontinuation due to adverse event was actually pretty comparable between the two arms. There's a lot we don't know about lorlatinib yet: resistance, overall survival, sequencing, and the implications of using lorlatinib in the first-line setting. But you could really make a compelling case today that lorlatinib really represents a new standard of care.
You brought up a lot of really great points, especially with how we're going to try and potentially tie this in with now frontline standard care of alectinib and also having frontline brigatinib available. So great points. Thank you so much. There is another trial. This is a combination study of apatinib and gefitinib in a phase 3 study of patients with EGFR mutant non-small cell lung cancer. So could you perhaps highlight what do these data show and how could we best sequence a therapy like this should it become available?
You know, apatinib is an orally bioavailable small molecule VEGF receptor 2 kinase inhibitor. And so this randomized study combined apatinib with gefitinib, a first-gen EGFR TKI, compared to gefitinib alone. Apatinib did improve outcomes. We saw an improvement in progression-free survival from 10.2 with gefitinib alone to 13.7 with the combination. That's hazard ratio of 0.71. So I would consider this combination an improvement over gefitinib alone, and is in many ways similar to the RELAY study that showed that the addition of ramucirumab, a monoclonal antibody also targeting VEGFR2, improved PFS over erlotinib alone. The challenge here is that our first-gen EGFR kinase inhibitors really aren't our standard of care. We know that osimertinib, the third-gen kinase inhibitor, is superior in terms of PFS from FLORA, even in terms of OS. It is better tolerated, and importantly, it has better CNS penetration, which is of particular importance, not just in treating brain metastases, but in preventing them. So should apatinib become available, I'm just not sure it finds a place in our treatment algorithms today since osimertinib is available here. But I'd certainly be interested in a combination of apatinib and osimertinib. And I think that would be worth exploring in the future.
Very interesting. Very good points. Thank you. Now, the Adora trial, that was one of the most talked about studies at the 2020 ASCO Virtual Scientific Program. And at ESMO this year, we're getting some additional results from this study in area of sites of disease recurrence. So what do these data tell us?
You're right that it was certainly the most talked about study. We're still debating the data we saw at ASCO, where we learned that adjuvant osimertinib after resection improved disease-free survival significantly at 0.17 hazard ratio. At ESMO this year, we saw results from a pre-planned exploratory analysis that detailed those patterns of recurrence. And we knew that recurrence would be less likely with osimertinib. It was 11% versus 46%. But here we focus on certain sites, again, focusing on the CNS due to the tropism for brain mets, because osimertinib offers neuroprotection as a highly CNS penetrant kinase inhibitor, and because brain recurrence can really be a devastating event. What we saw with this particular analysis was that CNS recurrence was much less likely with osimertinib. Only 1% of patients receiving osimertinib had CNS recurrence, compared to 10% with placebo, probably a bit higher than people realize. The CNS disease-free survival hazard ratio was 0.18. The 2-year CNS disease-free survival rate was 98% with osimertinib, 85% with placebo. Now, there were still some questions. While the study did require CNS imaging at entry, which was good, it did not specify the modality. And we know that MRI is much more sensitive than CT. While this may reflect real-world practice, it is still potentially a confounder if the use of MRI was imbalanced between the two arms. We also still don't know the impact on survival and whether the use of earlier osimertinib after surgery is any better than waiting until the time of recurrence, because osimertinib is still very active at the time of salvaging. And if it's really curing people or simply delaying recurrence, we just don't know the answers. We'll have to continue to follow up as these data mature.
Yeah, I definitely think that we're going to be talking about Adora for quite a while as a lot more data mature and we get longer follow-up. Now, KRAS is a target of great interest across oncology, but especially in lung cancer. And there are some further data seen with AMG 510 in patients with advanced non-small cell lung cancer. So what potential do you think that this agent has, especially after seeing the analysis presented at ESMO?
Yeah, a lot. I mean, this is, as you mentioned, a really important study. This was the CodeBreak 100 trial for AMG 510, now called Soteraspin. The response rate was 32%. And some people are already reacting, feeling that may be a little disappointing. You're coming from a context of targeted therapy where we have highly selective ALK, RET, EGFR inhibitors, where the vast majority of patients get a response. But this is really a different animal. These are heavily pretreated patients. And the comparison here isn't other targeted drugs, it's chemotherapy, which offers a response rate around 20%. So 32%, clearly a step up. And with Soteraspin, the responses were quick. They were durable with a duration of response over 10 months. And similar to targeted drugs, it was extremely well tolerated. Only one person stopped due to adverse event. To me, these data are compelling enough that this immediately becomes a preferred second-line option. Response rate's probably not high enough for me to use it in the first-line setting as monotherapy. But again, because it's so well tolerated, it'll pair well with other agents. So I really look forward to frontline combination studies, which are already underway.
Great, thank you so much. Now, tomorrow we're going to hear about the Empower Lung 1 trial, which is looking at cemiplimab monotherapy in the frontline setting versus platinum doublet in PD-L1 high non-small cell lung cancer. So could you discuss this trial and what we could potentially expect to hear from the findings?
Yeah, cemiplimab is an anti-PD-1 antibody, and this study, Empower Lung 1, was a randomized phase 3 trial to cemiplimab monotherapy or chemotherapy with crossover. Similar in design in many ways to Keynote-24 with pembrolizumab. What we saw was the response rates was higher with cemiplimab at 36.5% versus 20.6%. But most importantly, cemiplimab did improve survival. Overall survival with cemiplimab was 22.1 months versus 14.3 months with chemotherapy. It's a hazard ratio of 0.68. And if we look specifically at the PD-L1 high group, the impact was greater, with an overall survival hazard ratio of 0.57, despite a pretty high crossover rate in this study at about 74%. So clear efficacy with this compound, pretty consistent with other studies. So in a world where we have so many existing options already approved for immunotherapy in lung cancer, it's hard to see exactly how this changes practice, but I think sets the stage for combination studies going forward.
Good. Sounds like there's going to be a lot of combinations coming up on the horizon. I like it. Now, there was a very interesting international study called Lung Art. This is evaluating post-operative conformal radiotherapy, also known as PORT, compared with no PORT in patients with completely resected non-small cell lung cancer and mediastinal N2 involvement. What do you think is the importance of this trial?
This study's been going on for a little while, and we've been waiting for the results. And I think this really has the potential to change practice. Adjuvant postoperative radiotherapy, or PORT, has shown benefit in patients with resected lung cancer with N2 involvement. That's a subset where recurrence rates still remain unacceptably high. The data we have for the use of PORT were flawed. They weren't perfect prospective data, but a lot of us, including myself, had incorporated PORT into our standard treatment algorithms. Now, this trial was a randomized phase 3, simply looking at PORT or no PORT for N2 disease. It was a large, well-powered study, over 500 patients. Over 90% had PET, so good staging practices. And with a median follow-up of almost 5 years, we saw the disease-free survival about 30 months with PORT, about 23 months without PORT. That's a hazard ratio of 0.85, which did not meet significance. The 3-year survival rate, 66.5% with PORT, 68.5% without. No difference there. Now, there's a lot of nuance to PORT and its delivery, but this is an important study, and it really calls into question the role of routine postoperative radiotherapy for completely resected N2 lung cancer.
Very, very interesting points, and still we're learning that even if we hear some of these more negative trials or ones that are not showing too much of a difference, it's so important to be able to get these results and learn how they can apply to your clinical practice. So thank you for shedding light on this. Now, also in a more localized setting, there are data looking at neoadjuvant immunotherapy in lung cancer. We saw some preliminary results from a multi-center trial evaluating neoadjuvant durvalumab in resectable disease, and then also another one of neoadjuvant atezolizumab in the phase 2 Princeps trial. We'd be able to kind of shed light on both of these studies.
I think they were pretty consistent with studies you've reported on in the past, over the past year, Tina. We saw early results from the neoadjuvant IONESCO study. That was 3 doses of neoadjuvant durvalumab monotherapy for resectable non-small cell lung cancer. The response rate with durvalumab was about 9%, but the major pathologic response rate after surgery was double that at 18.6%. And again, we've seen this before. There's a disconnect between what we can see on the CT scan and what we see after resection. And that's really important to remember, that principle going forward, because some cases may even look like progression on a CT scan or a PET scan. At the time of surgery, there may be very little viable tumor remaining. Here, the MPR rate was pretty consistent with prior studies of monotherapy. This study was stopped early due to an excess 90-day mortality, which is a little concerning. Parsing through those data that were presented, it did not appear to be related to durvalumab or toxicity. It seemed more related to patient comorbidities, which reminds us that patient selection really critical to safely do surgical and neoadjuvant studies. The second study you mentioned, the Princeps study, presented by Ben Bess, was a phase 2 study of 30 patients. What was different here is they only received a single dose of atezolizumab after surgery. Again, very consistent with IONESCO, only 2 responses radiographically, but 4 major pathologic responses. Again, that disconnect between radiographic and pathologic responses. And metabolic responses in this study, very difficult to interpret. I think what we'll learn most from the Princeps study, really the correlatives, looking at the impact of a single dose of atezolizumab on the microenvironment, on immune constitution, and we'll see those results leak out over the next year.
Thank you. Now, what additional research being presented at this year's meeting, perhaps some that you might be authored on, would you like to highlight?
There's actually quite a bit presented at ESMO this year for lung cancer, a pretty full year. I had the opportunity to present data from the phase 2 RAIN 701 study. This is a trial that explored the activity of tarloxatinib. That's a rationally designed molecule that's given as a prodrug. And under conditions of hypoxia, such as those found in tumors, it undergoes a single electron reduction and fragments to an active drug that's extremely potent, an irreversible pan-HER kinase inhibitor with sub-nanomolar potency against EGFR, HER2, and HER4. This trial explored Tarlox in various cohorts, and we presented data in an oral presentation at ESMO showing very promising activity in HER2 mutant non-small cell lung cancer with an extremely favorable toxicity profile. Unlike other kinase inhibitors, this prodrug design really decouples the efficacy with the traditional dose-limiting toxicity at wild-type EGFR. The drug is also being explored in cancers with NRG1 fusions and EGFR or HER fusions. That's an important new subset of cancers. And again, study open to enrollment, actively ongoing. I was also able to give an update from Empower 133, showing that the addition of atezolizumab to chemotherapy for extensive stage small cell lung cancer significantly improved the likelihood of long-term survival. But in an in-depth analysis, PD-L1 expression, tumor mutational burden, those did not help identify who those survivors were.
Dr. Liu, thank you so much for sharing all of this fantastic insight today, especially during a very crazy ESMO schedule. We really appreciate it.
My pleasure. Thanks for having me.
That's all for today. Tomorrow on OncLive News Network, On Location, we'll speak with Dr. Priyanka Sharma for some valuable insight on the top breast cancer studies, and Dr. Yelena Janjigian for some expert perspective on pivotal gastrointestinal cancer trials. Thank you for watching OncLive News Network. I'm Gina Morrow.